It's on here.
Okay, so yeah, I'm talking about models in wound healing, scar tissue, and contraction. So I have a short attention span, so it's sort of really 3 shorter talks, which are sort of loosely coupled, and I am from Brigham Young University. So here's sort of the outline, so you know where. So I'll give a little introduction about scar tissue formation, and then I'll talk about some biochemical models, which actually could be force-based, but they're sort of just, I think of them more as biochemical models, which are sort of old. And then I'll talk about fibroblast populated collagen lattices, which means I need to talk about myofibroblasts versus fibroblasts, and the mechanisms for contraction of the lattices, and then some force-based models of contraction. And so it's sort of this part of the talk, then this is sort of a separate piece, and then this is sort of a separate piece. And this, of course, is the most recent and what I'm interested in more. Although I started out with scar tissue formation and sort of biochemical remodeling of the wound environment, and then I got interested in the forces and how important forces were in that process. And I've sort of I eventually would like to join the two, but of course I'm very slow and get distracted easily, so I've not got back to joining the two, but maybe someday. Okay, so of course, so all this is with dermal tissue. So in the dermis, if you wound yourself, there are several possibilities, but 2 of them are that you can repair the wound where the tissue is identical to the tissue that was there before, and so you can't distinguish the new tissue from the old tissue, which is sort of regeneration, or it can be replaced with tissue which is distinctly different from the surrounding tissue, which is scar, and this is typically what happens, and it happens to varying degrees. And so the question is, why does that happen, and what's involved in that? So here's some pictures. So the top is rat skin, rat dermis, and the bottom is human skin, I believe. And this is normal tissue, and this is scar tissue. And these are bifringent patterns, and so the brighter color means denser, more aligned collagen, and the lighter color means less dense and less aligned. And these are actually modified so you could see this one because this is much dimmer than this is. And so in this scar tissue, there is less dense collagen and it is less aligned than in the normal tissue. Whereas in this one, you can see that the collagen is aligned and here it is less aligned. So for our purposes, we're going for alignment means scar, but people could argue with that. And so here's the typical sort of cross-section. So here's the top of the skin. This is the epidermis, which we will not talk about. And it is tightly packed with cells. And this is the dermis, which is what I'm interested in. And the major dry component of the dermis is collagen. And collagen is the main connective tissue in your skin, in the body. And fibroblasts are the cells which are responsible for mainly for producing and maintaining the collagen. And the dermis is relatively acellular, but there are some cells in it, whereas this is, mainly cells. So if you have a wound in your skin, lots of things happen. And one of the first things is a blood clot fills the wound, and that provides a provisional matrix, which is mainly fibrin, for cells to invade the wound region. And so there are lots of things. You have an immune response and various stuff, but I'm interested in the fibroblasts eventually move into the fibrin clot and replace it with collagen. And if you scar, then the collagen in the region is different from the collagen here. So it tends to be denser and, for our cases, more aligned. So that's what we'll think of as scar. And so the question is, why does that happen? So what do I want out of the models? Well, I'd like to identify important factors in collagen alignment, because that's what I think of as scarring. So biochemical or forces, or sort of how these relate to each other, although I don't know how they relate to each other because I've just done them individually. I want to understand anti-scarring therapies. I would like to understand wound contraction and just understand wounds and wound healing in general. Okay. So the biochemical models. So the biochemical models has 2 components mainly, although it'll get complicated in a minute because I want to understand anti-scarring therapy, which means I have to add a lot more. But for now, let's just think about the 2 main things, which are fibroblasts, which are discrete elements. So here's a fibroblast, and this isn't really embedded in a collagen lattice, but there's collagen around the fibroblasts. And this is collagen, this is a higher magnification. But the idea here is the collagen is a much smaller scale than the fibroblasts. And so the collagen the extracellular matrix is modeled as a continuum variable, whereas the cells are discrete. And there are 2 main interactions, but there'll be more coming up. So extracellular matrix determines the direction of the cell. So this is just a corrugated surface, and cells have been placed on it, fibroblasts, and they align on the corrugation, but this is well documented contact guidance. And if you have a fibrous extracellular matrix where the fibers are oriented in a predominant manner, the cells will invade that lattice in the direction of orientation. And so for the model, F sub I is going to be the path of a cell, of the ith cell. This is the velocity of the cell. It is the speed, which shouldn't be bold, times the vector, which is some direction. And this vector is the extracellular matrix, so it's C for collagen. So this is sort of going to orient the fibroblasts. Yes?
Are you assuming that the cells are physically attached to each other as well? Or
no, the cells do not interact with each other at all. Yeah, except for through a chemical sort of, there's no sort of physical. Well, you could say there's some forced interaction, but it's all long range. There's no sort of, I don't assume any sort of physical contact. Okay, and then the cells align the matrix. And so here's a picture that was drawn by Stopak and Harris a long time ago, where they sort of imagined that the cells, they saw these actually. So the cells have deep involutions, and in the deep involutions is where the collagen is formed, the fibrils are formed. And so you can sort of imagine as the cell's moving off, it's sort of forming collagen sort of in the direction that it's moving. And so here's how it's modeled. Theta C is the orientation of the collagen. Theta F is the orientation of this vector F, which is a sum of the cells which are doing the reorienting at that location. So some weighted average, because this is the direction of the cell, although there's a little bit of time lag here. And so this is how the cells reorient the lattice. And so the basic idea is this. You just have a cell which is moving and it's directed by the fibers, but it's changing the fibers and reorienting the fibers as it moves.
Is it also degrading the collagen?
Yeah, but so far I'll talk about that now. So initially we started out with just sort of no net change in density, but now I'll talk about change in density.
In the cartoon there was...
Yeah, I mean, the cartoon was that way. So to really talk about anti-scarring therapy, I had to add a lot more. So we started out with some very simple things. But so now now the vector field, which represents the extracellular matrix, has two components. There's collagen and fibrin, because this is a blood clot. So B for fibrin, but B for blood clot. And the density is the length of the vector tells you the density of the protein. Okay, so the density can change by looking at how many cells are remodeling in that region, and then it can produce collagen and it can degrade collagen, and it can only degrade fibrin. Okay, the cells can only degrade it, and then they align it as before. They only align collagen because they don't produce, and they don't align the fibrin. And this is the same as before. I don't think I talked about that. So the theta C is the orientation of the collagen. Theta F is the orientation of the vector, the sum of the cells doing the reorienting. And if those are the same, then this is 0, and of course there's no reorientation. But if they're up to 90 degrees, then you get the most sort of reorientation. And this kappa is sort of an important parameter, but I won't talk much about that. Fibroblasts are allowed to proliferate, and then the extracellular matrix determines the velocity of the cell. And so in this case, it will determine not only the speed, because cells, if they're in very dense collagen, they move more slowly. If they're in collagen which isn't dense enough, I mean, it's sort of a bimodal thing. If the density of the extracellular matrix is too low, they don't move as well. If it's too high, they don't move as well. And so the speed depends upon the density of the collagen as well as the density of the fibrin. And then the direction is dictated by this vector V, but let's go to U. U says, this is the orientation of the collagen, and this is the orientation of the fibrin. So you take some linear combination of those to figure out U. That's sort of the direction the cell wants to move because of the extracellular matrix, the orientation of the extracellular matrix. And then you put U in up here, and this is a sum of the direction it wants to move according to what the extracellular matrix is telling it, how it's telling it to move, and the direction that it has been moving in the past. So this is sort of a polarization. So basically the cell's moving off in this direction. It senses that it should turn around. It won't, just change its leading edge. It will sort of veer off in a different direction. So this sort of indicates that it doesn't like to change its leading edge. So it has some propensity to continue in the direction that it was going before. Okay, and so then this is the V vector that tells you the velocity of the...
But why do you need the delay?
Well, if it's at the same time, then these paths don't... so that you can change... so we've done simulations as we change tau, and they're sort of an optimal tau. It depends on what you want, but as you change the delay, then you get of course different patterns. But you can imagine various things. So the cell sort of takes some sort of time to respond to its environment. So if it's sort of moving off in this direction and it decides to change, there might be some propensity to continue moving, and then it changes a little bit later. Or there are various reasons why you might want to put the tau in. So, but if it's 0, then
The direction V here is some interesting combination of dependency on rho and delta.
Yeah. So this is just how much you weight. If the collagen directs it more than the fibrin, or if the propensity to move in a continuing the direction it was moving in before, as opposed to move in whatever the direction the orientation is telling it to move in now. So for instance, if you have a very random matrix and this tau were very small, then the cells will sort of re-randomize the matrix, and you don't change the orientation much. But if the tau is larger, then I haven't looked at this for a long time. So if the tau is larger, then you'll get more orientation. But if there's a lot of alignment and tau is very small, then the cells will just sort of continue going in that direction, and they don't reorient the matrix very much. But if there's a larger tau, then there'll be sort of more. But then if it's too large, so you can sort of vary tau and see what happens with it.
So there seems to be a interconnected process in which the cells are pulling on the matrix, organizing it, and then migrating, and then pulling as well, right? So where is the force coming in?
There's actually very little. So you can imagine the force maybe is some weird, it's not explicitly stated, but it's sort of in here somehow. This is the effect of both biochemical remodeling and force remodeling, but it's not explicitly in here at all. And that's what I became more and more interested in. And so late in the end of the talk, I'll talk about things which are just force models of just force. So right now there's sort of, you can just say, well, this.
So these are essentially rails that you're going on right now.
Yeah.
In some ways.
Well, whether they're rails or not, yeah, sort of. I guess. Right. Well, I'm not sure that that's true. Is that accurate? So the fibrin matrix is random, right? Because the blood clot is just it's a random sort of polymerization. But the question is, if you put in a very structured matrix rather than a blood clot, would you get a very different pattern in terms of wound healing? And I think you would. So I don't think they're independent. So while it is true that the fibroblasts do not produce the fibrin, they degrade it, and the fibrin to some extent dictates how they move into the wound region. Sure. Yeah, no, something like that could, yeah. Although that wouldn't be force, but the force could, yeah. Okay, so here are the sort of pictures you get. And so here, this is the top of the wound. Think of that schematic before. The top of your wound, the epidermis would be up here. This would be normal tissue. This would be the basement membrane. So this is going deep into the wound. This is the wounded region. Initially, there isn't any collagen. There's just a fibrin. And then the cells invade from the side and the bottom, and they replace the fibrin with collagen. Now, the density is dictated by the color. So high density is red, low density is black. And then these are streamlines of the vector field, but you can think of them as sort of the orientation of the collagen. So I think of scars as being oriented collagen. And this is the placement of the cell. This is the distribution of the cells at the time at the end of the simulation. These graphs show you, this is vertical distance from the basement membrane. So 0 is down here, and it's an integral along this line of the density. So the density is high down here, which there's a lot of red down here. So you have high density. And then as you move up towards the top, which is moving this way, up here you have less density, and that's why the density decreases. And this just tells you the time course of the cells, number of cells in the simulation. So it sort of grows exponentially and then caps at the cap that the simulation allows it to go to. And so then you can see pictures like this, and hopefully this one will play. I was having a hard time earlier. And so this is the blood clot. This is the region. And I'm not plotting the fibrin. And so the cells move in. I'm not plotting the cells either. The cells move in and replace the fibrin with the collagen. And then here you can see some of the cells are given in this simulation, not all of them. And they sort of move in and
Why
replace the
are
fibroblasts.
most cells at the bottom?
Most of them do come in from the bottom, so various simulations have been done, but they believe, although it's not clear, but people believe that most of the fibroblasts come from the basement membrane as opposed from the surrounding tissue, but it's really not very many people have measured it.
I don't remember where's the basement. Basement membrane?
Yeah, down here, just from the subcutaneous tissue or whatever, what's under the stuff under the dermis. They believe most of the influx comes from here, but it's not clear where most of the cells come from. But that, some people have the only measurements I could find indicate the most. I mean, they labeled cells from they were trying to figure out which ones came from the local region and which ones came from the bloodstream, or I'm not sure what, but they had labeled cells and it looked like most of them came from below. That's what they were. But it's not very clear. It's not well known. So the influx of where the cells come from could actually be extremely important in terms of the simulation. Okay. So I want to understand the effects of TGF-beta, and so this was quite a while ago when TGF-beta was quite, it was a hot sort of thing, and so they had done, there was fetal tissue and adult tissue, and in fetal tissue there wasn't any TGF-beta, and in adult tissue there was, and so this was an anti-scarring therapy thought at the time. And then they discovered the different isoforms of TGF-beta, so it got very complicated. But TGF-beta1, less TGF-beta1 meant less scarring. More TGF-beta1 means more scarring. More TGF-beta3 means less scarring. And there's a TGF-beta2 as well, but I won't talk about that. And so the question is, what does TGF-beta do to the environment? Well, it does lots of things. So these are the effects on fibroblasts. It's a chemoattractant, but it's biphasic again. So at low concentrations, it causes them to come towards it. At higher concentrations, it sort of suppresses motion. It increases collagen production and decreases the degradation of collagen. It regulates cell proliferation. It regulates cell proliferation again in a biphasic manner. And it inhibits matrix degradation. So the main difference between the three different isoforms of TGF-beta are the different, all the graphs are sort of qualitatively the same. It's just the parameters. The peaks are at different places, but they all sort of have the same shape, which makes it kind of difficult to understand why this would have the opposite effect of this. So we want to separate these things out. And so here's sort of the simulation that you do to separate out. So if you say no TGF-beta, which should be good, not scarring.
Where is the TGF?
Pardon me?
Where is it?
It's uniform. It's uniform everywhere in the wound region, yeah. So there's no sort of, that may not be true, but so the wound region, this is like a millimeter deep and half a millimeter is sort of what I'm thinking of as wound region. But the only measurements on TGF-beta are time course, and there are very few time course measurements of how it varies over time in the wound region. And I could find no measurements of how it varied within the wound region, so I just assumed it was constant within the wound region. And so here I'm assuming there isn't any TGF-beta. Here I'm assuming it has an anti-proliferative effect. Here it changes the motility, and here it changes the production of collagen. And so then you can look at the different effects, and I say alignment is scarring, so you have a lot of alignment here, but not a lot of collagen, a lot of alignment. And you have sort of less alignment in this one than any of the three, but this would have been adding TGF-beta, and so that would have been counter to TGF-beta1. TGF-beta1 is supposed to produce more scarring, and here it looks like it's producing less.
If I superimpose the last two ones, where motility can also potentially change collagen itself.
Yeah. Yeah, and the density of the collagen actually changes the motility.
That's
too,
right.
So this is, so you want to add these effects together, certainly. So we do. And so you add them together. And so here is normal, which means TGF-beta1, and here is no TGF-beta1. And again, no TGF-beta, it's a little hard to say. This seems maybe, down here you have a sort of a clump of collagen, which is bad, a dense collagen, but you have more a randomness than here, so it's a little unclear. So it certainly didn't explain it.
Just the comment earlier was that the influx of where this begins has a determining effect, essentially.
No, yeah, it could be very important. So you can change
And this alignment that you've described is vertical.
Yeah, so because
if
you have a lot of them coming here.
Let me just make a point.
Mhm.
So if you are injecting more particles from the bottom than from the sides, you essentially a priori have a
Biasing
alignment or biasing.
Certainly.
So anything that suppresses the inflow or movement upwards in your simulation is going to get rid of the scarring, yeah?
Maybe. I mean, I don't think it's that simple. I mean, it will have an influence on it, yeah. So this sort of was unclear what was going on. It didn't explain the anti-scarring properties. So we went looking for something else, and so there's one thing that mentioned that TGF-beta1 influenced mesenchymal cells by having them produce more lamellipodia, versus TGF-beta3 cause them to have more philopodia. So if you think philopodia means they can sample their environment better, maybe reorient more frequently. Lamellopodia means they can't sample their environment quite as well, they sort of persist in their direction. Then you can run a simulation where they reorient less or more, and then you could maybe explain the effect of TGF-beta in the scarring and its anti-scarring therapies. And so that's sort of what we learned from, well, we learned other stuff, but that was sort of what I wanted to talk about here is we gave some insight into alignment and a better understanding of the TGF-beta in the wound region, or possible reasons why it works. And so, yeah, so that's sort of what we did there. Yes,
Alignment in your modeling is, I mean, you had a slide at the beginning that where the alignment is this way. And is that the alignment in a scar?
Okay, so in that scar it was, but it's complicated. So if you look at the
I'm
rat,
not seeing alignment in that direction.
yeah, no, if you look at the, yeah, that was a burn wound. And in the rat, there's less alignment. Well, the one you saw before, it was more random. But typically what happens with a rat wound is there's a dense clump of collagen at the base, and that's what we were seeing. And then it depends upon who you talk to as what means scar or not. So if someone looks at a tissue section, they'll say, yeah, that's scar and that's not. But whether you get agreement on exactly how alignment works is the more challenging question. So, yeah. Okay, so now so as we sort of did all of that, I became more interested in forces. And so I became interested in fibrillous populated collagen lattices. So I want to tell you a little bit about those and then what I've done to model those. So of course, fibroblasts populated collagen lattices have collagen, they have fibroblasts. And so they were fibroblasts were first grown on collagen by Ehrman and Gay in 1956 because they thought this was maybe better than growing them on plastic. And in 1976, Ellsdale and Bard were the first to report that fibroblasts would contract these lattices. And of course, wounds contract, and so this is an interesting thing. And Bell in 1979 and colleagues were the first to produce the fibroblast populated collagen lattices like they're produced now. And they also produced, a few years later, what they wanted to be a dermal equivalent, which never sort of worked out. But that basically means their fibroblast populated collagen lattices, they just made sure the density of the collagen was sort of equivalent to density in the skin, and the density of the cells was sort of equivalent, and sort of some of the properties were similar. But it never really worked out. But the fibroblast populated collagen lattices have been widely studied since then. And so why? Why do I care? I'm sort of self-centered, so maybe why you care might be the same reason why I care. But I care because, well, as I said before, on plastic, fibroblasts have a very different morphology on two-dimensional substrates as opposed to in a three-dimensional matrix. So here they're very spindle form-like, and here they're flat and spread. And so, of course, the dermis.
Help me with the pictures. So you're seeing that's grown on a two-dimensional substrate.
Yeah, these are just on a petri dish. So this is the bottom of the petri dish, and they're just flat, and they spread out and become very spread, and their internal structure is different. Here,
the image for me looks three-dimensional. Isn't this?
no, well, so there's some, there's some focus of, but it's so you're looking up from below and just focusing in on whatever focus plane you're focusing in. But I mean, they're three-dimensional structures, but they're pretty flat. Here, it's in a three-dimensional lattice, and
What is yellow and blue?
so green is actin and red is alpha tubulin.
Yeah. What is yellow and blue?
Yeah, yellow is the so there's just green and red, and these are just different mixtures of them. So that's where the two are superimposed. And then, the blue, sorry, is the nucleus. It's a DAPI stain for the nucleus.
On the right, every cell should be red, of course, because every cell should have tubulin.
Yeah.
There are many cells which do not have any red color.
I don't know.
Strange.
Yeah, I don't know.
They should.
Okay. So, the fibroblasts populated collagen lattices are a good system to study cell extracellular matrix interaction, and they can help us understand wound contraction. So this is typically what's done. They make these lattices, they add pharmacological agents to them, they see how much they contract, or they see what the difference is in how the cells are interacting with the matrix. And so these are widely studied for these reasons. Okay, so here's what they look like if you want. So these are in cloning towers. So these aren't usually done. These are very thick. And they turn into these little pellets. But this is typically what's done. You put them in a 24-well dish. It looks like this, and then several days later, it looks like this.
What are these dots?
Those are bubbles.
What is that?
They're just bubbles.
What's the scale bar of the bottom ones? Is it the same as the top?
No, this is a future. I don't know how well. There's your
So
thing,
the
but this is,
contracted one on the bottom right.
oh, these two are the same scale. So this is days before this. So this is a few days later. So this, which filled the well, is now that smaller.
And I'm assuming it's now floating as opposed
Yeah,
to.
no, they're floating in both cases. It's floating here. It's just they're, there's not much room for it to move.
Describe again the two processes. One is this build, and then later some reconstruction
This,
causing the contraction.
yeah, no, I'll talk about it. Yeah. So is there another question?
Why is it contracted on the right? On the bottom?
This is just after time.
After size?
Yeah, so the contract. I'll talk about that. So this is just showing that. So this is when they're initially cast, and then this is a day or two later, and they contracted. So there are two types of lattices you have to know about. There's free-floating lattice, and what you saw were free-floating. And they're attached delayed release lattices, which were done by Tomasek initially. And so these are done in petri dishes. And so basically what happens, collagen, when it is cold, is soluble, and when it warms up, it polymerizes. And so they keep it cool, they put the cells in it, they drop it on a petri dish, and it flattens out like a pancake, and then they put in the culture media, and it floats. And so this is the free-floating lattice.
Where are the cells?
They're inside the lattice. So they have the soluble collagen. Typically what it's done is you take soluble collagen, you add cells to it in solution, then you drop it on the petri dish, and then it polymerizes as it warms up, and then it floats in the media. Okay. And then the attached delayed release lattices, they do on tissue culture dishes, which the lattices stick to, so it's like a pancake on a griddle. You put the media in it, and the lattice remains attached to the bottom. And then several days later, they'll take a little spatula and lift it from the bottom, and then it will float. So it's attached to delayed release. Okay, so myofibroblasts versus fibroblasts. So in attached delayed release lattices, typically you have myofibroblasts. So I need to tell you about myofibroblasts. So myofibroblasts were first identified by Magino in 1971, and they were very excited because they were looking for what caused wounds to contract, and myofibroblasts exhibited characteristics of both fibroblasts and smooth muscle cells. And since smooth muscle cells contract, they thought, oh, this is great. This is what is contracting the wound. But it was not as simple as that, as it seldom is. So myofibroblasts are characterized by bundles of contractile microfilaments, extensive cell-to-matrix attachment sites, and intercellular adherens and gap junctions. But functionally, it's really this one. If you have a fibroblast which exhibits alpha smooth muscle actin, then they call it a myofibroblast. And then there are these other differences too. They have different cadherins, they have different integrins. So cadherins are how cells typically interact with other cells. Integrins are typically how cells interact with the extracellular matrix. Fibroblasts are more motile than myofibroblasts, and fibroblasts produce less force, and myofibroblasts produce greater forces.
What's the order of magnitude between those less and greater?
I don't know, but it is known. So off the top of my head, I don't know. So what causes fibroblasts to differentiate into the myofibroblast phenotype? Well, TGF-beta 1 will do it. Rigid matrix will do it. So this is why in attached delayed release, you put fibroblasts in this lattice, which is fixed to the bottom. They feel the stress because they're trying to sort of spread out on it. So they start to differentiate into myofibroblasts. And then this is sort of at low densities, if you plate them, then when they become confluent, they'll become you have more myofibroblasts, which is unclear why. What? Yeah, so this is just, if you plate them at very low densities and grow them to confluence, then you get more myofibroblasts than fibroblasts. Okay, so there are three mechanisms they believe for contraction, and for a mathematician, they're not that different, but for biologists, they're important to distinguish. So one is cell elongation, one is cell contraction, and one is tractional forces due to cell locomotion. And so here is cell elongation. So what happens is typically you trypsinize the cell, which means they round up, and then you put them in this soluble collagen, and then the collagen polymerizes, so you have these rounded cells in this extracellular matrix, and they start to elongate. And so this will cause some forces, they start to elongate. And this happens in the first 6 to 8 hours after fibroblasts are placed or cast in the matrix. And so if all your contraction happens in that time, it's mainly due to cell elongation. So cell elongation forces tend to be less than migrational forces, at least in terms of contracting the lattice. And so if you don't have very many cells in the lattice, then you don't see contraction in this early time. If you have really a lot of cells, then you'll see contraction in this early time frame. And it's sort of important for biologists to know this, because when you add different drugs to lattices, sometimes it looks like they're contracting a lattice. They prevent contraction. Sometimes they don't prevent contraction. And it's important to know the density of the cells in the lattice, because sometimes the drug will affect how they elongate or not. And so that changes depending upon which mechanism is causing the contraction. If it's this elongation because you have a high density of cells, then you get different results than if you have low density of cells when you add different drugs to the lattice. But for a mathematician, you would just sort of change the strength and the time scale.
Perhaps I misunderstood, but you're suggesting that in the cell elongating phase or regime, you get overall contraction?
Yeah, there's some contraction, but it's not very much unless you have a high density of cells. If you have a lot of cells in the lattice, then you get this contraction.
It seems counterintuitive in the sense that whatever it's connected to, this red fibrous network, it would suggest that that's getting pushed out. I'm somehow puzzled.
No, it's contracting, it's pulling it in. So here's the picture. So these are cells in a lattice. And so watch this one initially.
Just again, the word you used was the cell was elongating, so that's a suggestion that the cell is actually pushing out.
No, it's pulling to pull itself out. So so this guy puts out processes and grabs, and you can see it's sort of distorting the matrix around it. And if you watch this one, so let's go back, and you can see this one starts out rounded. It starts putting out these processes and pulling, elongating itself, and changing the lattice. And then eventually this one will sort of migrate off.
So these are on the surface or inside?
These are
the gel?
embedded in the lattice. And so actually if you watch them, some of them appear and disappear, and that's because they leave the focal plane.
So in this picture, cells should somehow assemble into, they attract each other.
No, they aren't attracting each other.
But in your picture, in your movie, they seem to attract and concentrate in one spot.
Yeah, I don't think that's...
And really, if they pull the lines.
Yeah,
If
they're
you
pulling.
want to keep going, they will do that.
They're pulling, but whether I don't think there's any propensity for them to aggregate necessarily. Here you see some of them aggregating, but I don't know that that's a...
But there's a lot of systems in which that does happen. You see it in endothelial cell cultures, for example.
And it's also true that if you have, if there's a cell here and a cell over here pulling and tugging, then it makes them you get a stress field between them, which is probably easier for them to move along that stress field.
They're very far. Their interaction will be very weak when they're far.
Yeah. Okay. So, okay, so the other one is cell contraction. So in this case, you have cells which sort of stretch out, and this is an attached delayed release. So they stretch out in the lattice, and the lattice is under stress, and so they're under stress, and then you release the lattice, and so then they sort of can crumple. Yeah.
I don't get the difference between contraction and elongation, because
Yeah. So
when the cell elongates, it has to. It's the same like contraction, because the cell expands, but it also pulls. Otherwise,
yeah, but this
you
isn't.
wouldn't get an elongated cell.
Yeah, no, but so this happens when you release this. This will happen immediately.
Yeah, because of tension.
Right. Yeah, it's because of the tension, whereas the other one is sort of active remodeling of the cell, sort of to stretch it out, to push it
Is
out.
there different terminology perhaps? Since there's a force concept here, so one is a pusher and the other is a pull.
Yeah, no, mathematically, it's not too different. In the sense here, you have stress, and then you release the stress and watch it sort of do.
Again, just sort of idealizing, I've got a unit, this cell, and one cell has the action of pulling on the surrounding matrix, and the other has the action of pushing.
Yeah, no, it doesn't. The elongation doesn't really push the matrix. It sort of reaches out, grabs, and then pulls, and sort of. Then it pulls itself, and that's how it stretches itself longer. It makes itself longer. Although internally, it's doing a bunch of, there's the cytoskeleton is being reorganized so it can stabilize this as it pulls itself and stretches itself out.
Well, myosin only contracts, but the other hand, you have osmotic pressure that can increase the volume of the cells. So what is actually working?
Yeah, I don't know exactly. You have to ask the biologist more about how they
Osmotic
elongate.
pressure will be constant in the system.
It is constant.
It's not changing.
But it is true. What is indicated because of the experiments that this elongation process is affected by different drugs in a different manner than, for instance, cell locomotion. And so if you don't understand, so if you distinguish the three mechanisms mainly for biologists, because if it's elongation which is occurring and you add certain drugs, they will affect that process more than they affect cell motion, and so you get different effects in the lattices when they're contracted, depending upon which is the dominant thing contracting the lattice. So here, in attached delayed release, you can see these sort of cells. So this is in a lattice which is attached and then stained, and this is right after it was released and then stained. And so you can imagine these cells at one time were stretched like this, but then when you release the lattice, these are sort of crumpled up.
What does release mean?
So the lattice is first attached to the bottom of the dish, and it stays that way for several days, and then you take a little spatula and lift it off, and then it's floating in the media.
Mechanical release.
Okay, and then the tractional forces. So this is a silicon substrate floating on something, and you have the fibroblasts, and so these are the wrinkles in the substrate. And so the fibroblasts sort of wrinkle the substrate as they're sort of moving on the surface of it, sort of going like this. Okay, so the model. So force space model. I have cells, I have a collagen lattice. So here's the cell, and so there's a center, which is the nucleus, it's the blue thing. And there are integrin-based adhesion sites which are attached to the center. They're the red X's. The green is just for your visual pleasure. I don't model the membrane. And then you can look at the forces of fibroblasts and figure out what they are, and so you can sort of figure out what the spring constant should be. And then the lattice is modeled in the same manner. You have these nodes which are attached by springs or viscoelastic elements. For our purposes, they're springs. And they don't have to be regularly placed as shown there, but they can be randomly placed as shown over here. So the intersection of the blue lines are the nodes, and they're connected by a Delaunay triangulation. And the cell adhesion sites, so the red X's before, can interact with the nodes, but the nodes only occur where you have blue lines intersecting. So here you have the green stars, which should be red X's, which are interacting at intersection of the blue lines. So this is the cell. These are the adhesion sites. They're interacting with those nodes. And
Why they're only attaching to the nodes?
because they're just allowed to, they only interact with nodes. That's all I'm allowing in the model formulation.
Just in the model, but there's no.
That's right. There's no reason for that. Just
No, I mean, even in reality, they attach to particular sites on the collagen fiber, right? So one can imagine that these sites are randomly placed where they are distanced. They're not sort of on the
Yeah,
entire fiber, right?
but-
So there's a particular sequence on which they attach.
Yeah,
They're
but
not
it's-
nodes.
But it's certainly more, the nodes are way more sparse than that sort of thing.
But it is specific.
It would be a discrete
Yeah.
sort of thing.
I mean, maybe there's a length scale issue.
Yeah.
How large is the cell scale relative to this?
Yeah, so the
node
cells, when they're
separation?
elongated, can be like hundreds, like 100 microns long of
I mean,
fibroblasts.
relative to the scale.
Yeah, so the fibroblast, on the scale, the nodes would be extremely close, you know.
So it's a continuum, essentially.
Yeah, so they really probably should be able to reach anywhere along the collagen fiber. But I'm just doing this way. Okay. So then you can do the Young's modulus for collagen lattices. There's one other thing. So as two fibrils come together, collagen fibrils come together, they will polymerize. And so to model and form a larger bundle, a larger fibril. And so to do this, I increase, if two nodes come together, then it's not cross-linking. This happens without cross-linking. So this is just a natural, if you take two fibrils and bring them close together, they will form a larger fiber together. So they come together, it's a self-polymerization sort of process, which isn't part of cross-linking. Yeah.
Hydrogen bonding. Oh, super.
So what I'm intrigued here is the Young's modulus for a bulk, which is not what the cell is seeing, right? The cell is seeing the stiffness of an individual fiber.
Yeah.
Which is different
Sure.
in terms of its viscoelastic properties than the bulk, for a variety of reasons. Cross-linking is one of them,
Sure.
bonding is another.
Mhm.
So how do you sort of bring the continuum level measurement that goes all the way from 4.5
Yeah,
pascals
and-
to a kilopascal to a fiber which may actually be very stiff or not?
Yeah, no, that's a valid point. And so we optimize over the modulus for the collagen. That's one of the parameters we vary because There's a huge, well, it depends upon the density of the collagen too for the bulk. I mean, it's true. That's
Just to understand that, Mark, you're suggesting there's a distribution of strengths within these
Well,
fibers.
So yes, and also the fact is that these fibers, depending on how long they are, how cross-linked they are, will respond differently to tension and strain. Right. So it's a very inhomogeneous network in terms of its size, in terms of its pores, in terms of its bundling. Imagine if you have a rope that is this thick versus a rope that is very thin. Same building block, but the cell is seeing the rope, not the entire matrix, because at the length scale of the cell, you're seeing only the thickness of that particular piece of rope. So it's kind of
Well, they're also non-linear. This elasticity of this.
collagen is never homogeneous.
right. Yeah. I mean, there's a,
You cannot do anything, everything.
Maybe, I don't know, because it can also walk on a single fiber, or it may be very dense. Anyway,
okay. So here's the equations, if you want to see them. So the x is the cell center, there I tells you which cell. So this is the velocity of the cell, is the sum of the forces. There's not any acceleration because of low Reynolds number. And so this is just the stretch of the springs, the spring constant, and the direction of the springs. This is the cell center, this is the node, and the PIJ says the jth attachment site on the ith cell is at the Y node. So there are Y, K are the nodes in the collagen. If you specify the appropriate K, there might be an adhesion site attached to it or not. And so that's how this moves, just according to you're summing the force of the springs. And but the nodes move according to cells which are attached to them versus other lattice sites which are attached to them. And so you just have those two.
Second, so the constant here, this is isotropic.
Yeah.
So the cell is reduced to a point.
The nucleus is a point. It has adhesion sites which reach out.
Following this, the nucleus center.
That's right.
And the constant C and little gamma, how do they relate? What's the ratio of size of these dissipations?
Yeah, so this is much smaller than this, but it's hard to know what would be. But I optimize over them. Yeah. So it depends upon what you want this to be. So if this is just a node, you're pulling, this comes back to sort of the bulk property of the collagen versus not. You have a node and you're sort of pulling it through this gel, what should be the drag coefficient, as opposed to a nucleus being pulled through a gel, or is it, yeah.
I'm just wondering whether or not you can just set the left-hand side of the second equation to 0 to make it essentially quasi-static.
The second, yeah, that might be a reasonable thing to do. Okay, so then, but with the polymerization, it's a little bit different because if the nodes aren't linked to each other, then there's no force. And so this is just if the d is the distance, the length of this vector. So if the nodes, they don't resist compression unless they are linked. So if you have a larger fibril, then it will resist compression. But if it's a small fibril, then it won't resist compression. And then this is just the spring. And if it's a larger fibril, because it's been linked, then you change the rest length and the spring constant, make them higher. And then this is the cell contribution. And so the only other thing you need to know is the nodes randomly reach out and find, you give some distribution, they reach out, and then they find the adhesion sites reach out and find the nearest node to that, and then they remain attached according to some distribution which is given. And so then you have this. Oh, no, you don't have that. So there's one other formulation we did, and that's because things weren't working out, so I thought, well, maybe it's because of the bad formulation of the lattice. And so we did a string formulation where you actually look at nodes on a string. So here you have a string, you can sort of follow the strings. So it's not like a triangulation. And now, so the nodes are only interact with two adjacent nodes. So this node is only connected to that node and that node, the two adjacent ones, because they're sort of beads on a string. And then everything else is sort of the same, although you can polymerize. So a node from this string gets close to a node from that string, they will link up with the spring sort of to bring the two strings together and form a stronger bundle. And yeah. So here are the pictures you can sort of see. So this is, if the lattice is fixed, so the blue is the lattice, the red are the cells, and so the cells just sort of move along the lattice. And here is just, I won't do that one because let's look at this one. So if you don't fix the lattice, then you can have the cells contract the lattice. And there's some sort of organization within the lattice, which is nice. And it's irreversible because of this polymerization where you change these spring constants and lengths. And then if you look at the string simulation, then you have this. And so you see the pattern's quite different, but you still get contraction. So,
with hexagons or something like that?
yeah, well, the big holes, I'm not sure if they're exactly hexagons. I haven't really looked at what happens, but it's much less connected than the triangulated. And if you put the nodes out and triangulate them, that's a much more connected network than if you just put a bunch of strings which are connected to their adjacent nodes.
The topology of that string network, do you permit it to change through the simulation?
So only that you can connect nodes which weren't connected before if they're from different fibers, so they will connect and polymerize. Okay, so here's the data I wanted to match, and it took me forever to match it, which is why we went to the string formulation. But so the boxes are data, and so this is low density lattice. So 3,000 cells, 10,000 cells, 30,000 cells, and 100,000 cells, and it's over time. So you watch the lattice as it contracts. So it starts out at whatever it is, and you see the percent of contraction over time. And so I wanted to match these. So the lines are the data, or the simulations. The squares are the data, the average of the lattices that were measured. And then we optimized over several parameters, and we're able to fit it reasonably well. And if you...
That's an exponential approach, yeah?
It's a what?
This is an exponential decay to the final state.
Yeah. Well, it's not, it's hard to, you can't just do an exponential. There's actually two, I'll discuss this in a second. There's sort of two things going on. There's something going on here and something going on in the other part where it's turned off. And so if you look at these, and so these, you'll see it here. This is the cells remain relatively fixed to where they are. They don't move very much. And that's because this is stress dependent. So to make this matching, I forgot to tell you the most important thing. Two things had to be added. The cells had to sense the stress, and cells are mechanosensing. So they sense the stress in the lattice, and if the stress was great enough, then they would remain attached longer, and they would have greater force and maintain their sort of shape, so they wouldn't keep contracting. And this is motivated because myofibroblasts have greater force. They're less modal, so they seem to remain attached more and more fixed. So adding the stress dependent allowed me to match the parameters. And you can see here that the cells didn't move very much. And there are regions in the lattice which are not really contracted very much at all. So if you look at the string simulation, you get the same sort of thing. And so the parameters here were varied just a little bit because the string simulation is much less connected than the lattice one where you triangulate it. And so when the cells have to stop the stress, according to the stress in the lattice, when they're become fixed, changes in the string simulation, but otherwise things are quite similar. So we were able to match it well there.
What stops the contraction?
If they sense a local high stress in the lattice, then they will remain attached. The attachment parameter will become longer, and they change their force, and so the rest length of the attachment sites is changed to be whatever it is when they are activated because of the stress, and they become stronger. So basically, they stop reaching out and grabbing, and they sort of become stronger and more fixed. The springs become stronger and fixed.
Because of the thicken.
Yeah. Well, the cells. This is the cells, not the lattice. So the cells respond to the lattice. So it's sort of, the idea is maybe they're differentiating into myofibroblasts. Myofibroblasts have larger contractile fibers. They're stronger and they're less modal. They sort of stop moving and sort of just hold things in place. Yes?
I wonder how many nodes did you use for representation of the sound? Because you have this constraint of attaching just two nodes of the network.
That's right,
So
yeah.
I guess if you would increase the representation of the cells, say, by a fold, that would change a lot.
Yeah, it's not clear to me if it will change that. It may, and I haven't varied that a lot, so I don't know. I can comment on that in terms of the motility on a two-dimensional sort of flat thing, but I don't know how it affects the lattice contraction. So here you can just see the different densities, and again, the point to notice is the cells contract near them, but there are large regions away from the cells which aren't influenced. And here's the string simulations. And so the thing, well, the string simulations, you can see part of the the way we check the area is the convex hull of the nodes. And so you get extra area on the string ones because you have these strings which remain out there. And let's skip this and this and this and talk about this. Okay, so this is what I wanted to match, was able to match, but it took me like 5 years to do it because I'm sort of stubborn. And if I did what I wanted to do, which is not have mechanosensing, if you use the same parameters that were from the other graph, you get this if the cells don't have any mechanosensing. And the point is, the cells can just keep contracting the lattice a lot more than they're supposed to. And so you can fit it better if you change your parameters, but you can only fit one, you can't fit all of them together.
But they're not infinitely compressible. You can't get down to 0 area.
Well, yeah, but you can get down to a tiny pellet, right? I mean, you can contract a lot more than 20% of the lattice. And so here's the simulation. And you can see here the cells are moving from their original location. They move out to a region and then sort of pull it in and then move out to a region. So there's a lot more motion of the cells. And so looking at the data, it's clear that there's a lot of contraction initially, and then the cells are sort of turned off. And so originally the question was, well, is it just the stress within the lattice, which is so strong that allows, so the cells just can't contract the lattice anymore? And the simulation indicated that was not the case at all. The cells had the capacity to keep contracting the lattice a lot more than they did, so there was something that was turning them off.
So
Yes.
could it be that they stop contracting because they are quote unquote talking to each other? So there's signaling going on, and at some point they realize, right? So it's a stop and go kind of a mechanism.
Yeah, but it's not clear.
When you,
I think it's all force-based. So the thing is, the signaling, it's not clear that there are any sort of, so you can have very low density lattices. You know, the low density lattice, this one, the cells are sparse in the lattice, and it's unclear whether they're connected at all. So what you mean by communicating with each other, if it's some chemical they're putting out or what, I don't know, but it's not clear that there is.
what's the
So it
average
would just be
distance in,
force.
let's say, black between cells?
I don't know that. But
Because I think the right experiment there would be to block their signaling mechanism in which they are communicating with each other.
well, they don't know if there is a signaling mechanism. They don't know if they're communicating with each other. There's no evidence, you know, if you talk to Paul Ehrlich, who did this all the time, he would say there's no contact and they're really not, you know, so at high densities maybe they are, but in general, he didn't think there was much going on with that. Yeah.
In your sensing model, how does the force of the individual cell evolve over time?
I don't know. I didn't look at that. These ones that I skipped show you sort of the force of the cell at the end time. So some are exerting a lot of. These have high stress, and these have low. And there's no sort of pattern. This is the radial. If you go out from the center looking at them, there's sort of no pattern. You know, there's not more stress in the center. But I don't know how it evolves over time.
You have aggregations where you have mixed forces.
Yeah. But this is what they believe happens in the lattice, that there's some regions of the lattice there's high stress and some regions which are less stress.
If you distribute your cell homogeneously within the lattice, how would this look like?
I don't know. I don't know if they could do that, right? I mean, because they just pipette it in, and so it's sort of homogeneous.
You can do it on the computer.
Oh, well, yeah, I could do it on the computer, that's right. So the point is the cells have a great capacity to contract the lattice more than they do. So what stops them? And so then this is true in the string simulation as well as the lattice formulation, the triangulated one. So if you just have them fix their attachments when they feel the high stress, they still contract really a lot, more than they should. Although you can make these fits better if you optimize. I'm always using the same parameter set from these pictures. So if you optimize with just this thing, but you can't fit all of them as well as you can when you have both mechanisms. And so then you can say, okay, well, what happens? Oh, but the string one is less dependent upon that. This fits much better. This works much better. And that's because there's less connection within the strings. So as the strings get pulled in by cells, if the cells become fixed, the strings are less connected to other strings. And so there's less contraction because it's a less connected network. But then if you just look at the stress-dependent strength, so if you change the strength and the size of the springs in the cells, you also get a lot more contraction than you should, so you need both. Although, again, the string simulation is not as dependent on that as that one. and we'll skip that and this. And so the conclusions are the lattice and string formulation give the same qualitative results for the contraction. The cell, there are a few differences, but the cells seem to need to be inactivated due to high local stress. Otherwise, they're able to contract this lattice a lot more than what the data says. And the cells only compact the lattice local to where they are, and this is consistent with what Gooch found in 2013. So he did some modeling and some experiments, and he said the cells compacted the matrix in the paracellular region much more than the average compaction experienced in the entire matrix. And then Stevenson, who did modeling only, they were trying to match data with different densities of collagen, not time data, and they could only match it if they assumed that the ECM compaction primarily occurs in the paracellular region. And so this is consistent with both of their works, in saying that the cells become inactivated, I think probably by force mechanisms, and so they just are able to contract the region near them and not continue to move off into other regions and contract it. Yeah, yeah.
What if there is a co-culture? The cells are of various different kinds, right? So you have multiple cells, and then what would happen in terms of the steepness of your curve, because different cells pull with different strengths.
Yeah, no, I don't. So fibroblasts I think fibroblasts do the best job of contracting lattices. I'm not certain of that. But when you put like mast cells or other things in it, then a lot of other things happen. And people have done that, but I don't know much about what goes on there. But like if you put, other cells usually don't contract. I think fibroblasts are the best at contracting lattices. Okay, that's all. Thank you.
Further questions?
So you started with this chemical model,
Uh-huh.
you moved to a force model.
Uh-huh.
What was some interpolation of the two? In some sense, I mean, this is almost linking to this question of chemical interaction.
Yeah.
But I'm sort of surprised that, I would be surprised if a force-only model was sufficient to understand this.
Yeah, well, so for the lattice contraction, there isn't much there's not much remodeling of the collagen. The cells don't produce much collagen. So these lattice contraction depends upon how long you leave the lattice. So if you leave the lattice for so I'm not sure, I may not be asking a question.
The word chemical in a slightly broader sense. So chemomechanical or thermodynamical, I mean, a force balance alone would not seem sufficient to model this type of interaction, because in some sense it's energetic as much as force. Force is only one aspect.
No, I think you're right in terms of wound healing, but in terms of the lattice contraction, I think force is probably reasonable. So these are floating in a media, and you can put several of them in the same media, and so there's diffusion through the full media. I mean, I don't think there's a chemical component I don't think the chemical component is very important unless it's bound to the lattice. But if it's if it's freely diffusing in the media, it diffuses through all of these, and I don't think that's an issue. And in the first few days, which normally they just look at these for 40, well, it depends upon which lab, but if you just look at the lattices for the first 48 hours, the cells don't do a lot of remodeling biochemically of the collagen. If you go longer than that, then they start producing collagen, and then things change. So I don't think there's much, for the lattice contraction, I think force models are a reasonable thing to just look at. Unless you put in other cells, in which case then maybe there's some sort of interaction with the other cells. But in the wound healing, yeah, then it's much more complicated.
Any other?
In your second model, can you predict something for the lattice? So, for example, if you reduce biochemically the force of the cells by blocking myosin or something?
Yeah, no, so what I've done so far, no, but this with the mechanosensing, you can predict distributions of like where myofibroblasts should be versus not myofibroblasts. And you could also, if you like inhibited myofibroblasts, and you could predict what would happen if you didn't have the mechanosensing. So if you assume that the ones that are mechanosensing become stiffer and less modal or sort of differentiating into myofibroblasts, you could add agents to prevent that from happening, and then see, so you could, in the model, you could prevent it. Although they have done, they've tried to prevent them from differentiating to myofibroblasts, and so those experiments are probably all done. So although we could, the model would be able to maybe see if our model matches those experiences. I don't know if it would, but there are a few things that you could predict in terms of the distribution of the myofibroblasts and stuff. And then what I'm actually interested in with scar tissue is sort of the pattern formation of the lattice when it's contracted and sort of looking at that. We just haven't looked at it. I've been mainly interested in how much it contracted.
Well, let's